Okay, greetings to all and welcome to the ZOOMinar today. I'm pleased to welcome the speakers for today, Dr. Thomas Michaels from ETH and Dr. Daniel Raleigh from Stony Brook. The first speaker, Dr. Thomas Michaels, studied physics and mathematics at ETH Zurich, then went on to do PhD in biophysical chemistry from the University of Cambridge, working with Thomas Knowles, and then postdoc in applied mathematics at Harvard University, working with Lakshmi Narayan and Mahadevan. And he was a lecturer in theoretical biological physics at University College of London. Then from this month onwards, he is an assistant professor of soft and living matter physics in the biochemistry department at ETH Zurich. His research is focused on the investigation of complex systems in biology, including protein aggregation and phase separation. Thomas, thanks very much for joining us today. Please get started when you're ready.
Well, thank you very much for the kind introduction and also for the invitation to be here and speak today. What I will do is I will try to give you a bit of an overview of some of my research activities, particularly in the context of understanding the mechanisms of protein aggregation with the goal potentially of devising strategies to control this process. So as has been said, I am a physicist, so I try to use ideas from physics to try to understand complex problems in biology. A lot of my work has been around trying to understand the various states that proteins can adopt, and in particular, this transition from the soluble state to the amyloid fibril state, and so the aggregate state, which is associated with a lot of diseases. But of course, we now know that this solid state is in fact connected with another range of protein states, including, for example, liquid condensates. And so my interest recently has also shifted towards understanding the role of this liquid condensate. And so one key topic of my research, and which was one word, keyword in my title, is this concept of control. And so let me try to introduce you to what is control. So control theory is a branch of mathematics. It's very applied in engineering, and it's related with concepts of learning and optimization. And so typically, when you have a control problem, you are considering a dynamical system, which is described in terms of some state variables, for example, the position of a particle over time. And on top of this, you have a control variable, which is an external variable that you can tune from the outside to drive the system in different directions. So for example, if you think about the position of a particle, the external control could be the force that we apply to the particle. And then depending on the protocol that we choose for the force, the trajectory will take different directions. And now the control problem consists of finding the optimal control, U, that minimizes a certain cost function. So the idea is that both the state of the system and the control may come with a cost or a benefit. And so what we want to do is we want to minimize the integrated cost over time. And this problem can be recast or formulated with control theory, and there is a set of tools, in particular Pontryagin minimum principle, which allows you to solve this type of question. This type of question is actually related with concepts of learning, in particular reinforcement learning, where an agent tries to optimize its action based on a reward cost function. So the actions that lead to a reward or that minimize the cost are going to be reinforced. And this type of reinforcement learning algorithm converges towards the optimal protocols that you can get from control theory. So maybe to give you an idea of how a control problem looks like, we can take the example of sailing. So imagine you want to sail from point A to point B against wind. So the direct path would, of course, be the shortest solution. But of course, this is not a viable solution because you wouldn't move. So what you do instead is you take a zigzag trajectory, which is called tacking. And in this way, you make the trajectory a little bit longer, but you gain in terms of speed. And so you minimize the time of travel from point A to point B, which in this context could be the cost function of the problem. And this type of strategy where a particular constant control is applied for a certain interval of time, it's called the bang-bang control in control theory, and we'll come back in later in the talk, as we will talk about protein aggregation. So this concept of control theory is very useful in context of engineering, and I have tried to apply these ideas in the context of protein aggregation, in particular in the context of understanding amyloid fibril formation. So I guess for this audience, I don't have to go to give too many details. So we all know that these amyloid fibrils are this general class of supramolecular structures that form from the assembly of proteins. They are characterized by this cross-beta sheet architecture, which is largely conserved. And so there is a wide range of apparently unrelated proteins and peptides that can form these amyloid fibrils. And the main reason why we are interested in these structures is because of their link with a lot of diseases, including Alzheimer's, Parkinson's, and type 2 diabetes. And so in this context, an attractive strategy is to think about inhibitors of protein aggregation. And so this could be small molecules or molecular chaperones or other molecules that are able to bind to some of the species along the aggregation pathway, and in this manner affect some of the molecular steps of aggregation. And now this idea is actually very much related with a control question, which is to say that this compound comes with a toxicity at high doses. And so that raises the questions of what is the optimal strategy that arises from a balance between the degree of inhibition that I obtain from this compound and the intrinsic toxicity of this compound. And in particular, how does this strategy depend on the molecular mechanisms of action of this inhibitor? And so to answer this question, I have combined optimal control theory with the kinetics of protein aggregation, and now I will tell you what I have discovered doing this. So the first step in the control problem, as I said before, is to set up a dynamical equation for the system, for the state of the system, which in this context means effectively understanding the mechanisms of aggregation. And so this is very briefly the way we have approached this problem, largely in collaboration with Thomas Knowles. And so the first step in this has been effectively to extend the concept or the tools of chemical kinetics to complex biomolecular pathways. And so we first start by writing down all the possible physically meaningful ways in which aggregates can form. So initially aggregates form by a primary nucleation step, whereby the monomers spontaneously come together to form the smallest stable fibril structure. And then these structures can grow by elongation. And of course, if we care about microscopic reversibility, we have to account also for the inverse process of dissociation. But primary nucleation is not the only way in which you can form new aggregates. You can form new aggregates also through fragmentation, whereby when a filament fragments, you have two filaments and so on. Primary nucleation forms aggregates directly from the monomers. In fragmentation, we form them from the aggregates alone. And so you may think that there might be also the possibility, if we want to be very general, to have a mechanism which involves both aggregates and monomers in the process of forming new aggregates. And an example of such a process is surface-catalyzed secondary nucleation, whereby the surface of existing fibrils can catalyze the nucleation of new ones. And then what you do is you write down kinetic rate laws for all these different steps that take into account the concentration of the fibrils of different type. And then you try to find solutions to these equations. I won't go into the details, but it turns out that you can actually solve these equations in particular limits for describing experimentally observable quantities. Such as, for example, the mass of aggregates that is formed over time. So that would be the total mass of aggregates, which is what is typically observed experimentally, for example, with fluorescence experiments. And so quite magically, it turns out that the solution to this equation is nothing but a generalized logistic function, which to biologists might sound very appealing because it bears a link with a lot of processes that have logistic growth, like bacterial replication and so on. And so in this context, there is effectively a fibril mass concentration, which increases as a function of time according to a sigmoidal shape, which is characteristic to these logistic type of processes. Initially, the concentration of aggregates increases exponentially with time. And then as we run out of monomers, the slope slows down and we reach a plateau. The multiplication rate in the exponential growth phase is this parameter kappa, which quite interestingly appears to be the combination of the rates of growth and secondary nucleation. It is actually the geometric average of these two rate parameters. And so this is very good because it means effectively that we can now interpret experimental measurements in terms of microscopic mechanisms, which effectively means that we can put numbers to these various rate constants and understand the relative importance of these different processes. And so here I'm showing to you a few examples of systems that form filamentous aggregates and that we have tried to describe using this type of theoretical tools, ranging from prions to actin in the presence of branching complexes to amylin. So what you see here are experimental measurements over time of fibril mass formation as measured by THD fluorescence, taken at different initial monomer concentrations. And the solid lines are the best global fits of the data to various models, in particular to this to this integrated rate law. And what this kind of fits allows you to do is to obtain values for the various rate constants and understand, for example, which one is the dominant mechanism of aggregate multiplication. And it turns out that for these different systems, the dominant mechanism of aggregate proliferation is quite different. It is fragmentation in the case of prions. It is branching in the context of actin in the presence of this complex here. And it is surface catalyzed secondary nucleation in the context of amylin. It is very important here to note that we are doing global fits as opposed to individual fits. So that means we are not fitting every curve with a different set of parameters, but we are trying to fit all the curves at the same time using a single choice of the rate constants and taking into account the variation of the monomer mass as dictated by the experiment. And so this is very important because it allows us to be overly constrained in our fit. And so whenever you manage to do a global fit to the data, it's actually mechanistically relevant. On the other side, it's very easy to do to fit data with individual fits. And so it's very important that you do this type of global fits. And here I'm showing to you another example from the group of Thomas Knowles about the amyloid beta peptide, which is one early example of the use of this type of kinetic tools to understand the mechanism. And this will be relevant for the discussion of amyloid beta that will follow later in the talk. So what you see here again are experimental measurements of aggregate formation over time at different concentrations, so 9 different concentrations here. And the solid lines here are the best fit to the data if you assume that the main driver of fibril formation is primary nucleation. So you see that the data are not captured by the model. So maybe it is fragmentation. Again, the best global fit doesn't describe the data, but if we think about a secondary nucleation model, then we are able to fit the data perfectly. But crucially, we've learned something about the mechanism, in particular, that there is an autocatalytic loop mediated by secondary nucleation, whereby the more fibrils I have, the easier it is to form new fibrils, which in the context of forming potentially toxic oligomers, it's not good news. And so, of course, the interesting species here from a disease perspective are the oligomers. And so Here, this is work together with Sarah Lindsay, who has developed a very nice technique to measure oligomer concentrations over time during an ongoing amyloid aggregation reaction. And this allows us now to ask mechanistic questions about the oligomers themselves. And so in the simplest scenario, we could imagine that these oligomers are simply short fibrils. So the only difference between an oligomer and a fibril is the size. And so in this case, you can write down the rate laws associated with this mechanism. And here is the best global fit of the fibril formation data and the oligomer data to this model. You see that we are able to fit the fibril formation data, but the oligomer prediction is off by 5 orders of magnitude. So something is wrong here. So what do we know about these oligomers? So we know that these oligomers have a structure which is different from the fibrils. And so we could include a conversion step from the oligomers to the fibrils. And so if we do this, again, write down the rate laws associated to this problem, here is the best global fit. We are able again to capture the fibril formation data, and the oligomer data are now captured in a better way. At least we are capturing the right order of magnitude for the concentration, but the time scale is off. So why is this? Well, it is because we have forgotten about microscopic reversibility. And so the fact that if we allow for oligomer formation, we have also to account for the reverse process of dissociation. And so if we take this process into account, now we are able to fit both the fibril formation data and the oligomer data perfectly, but crucially, we've learned something about the mechanism. And so we can now ask questions such as, for example, are most oligomers going to convert to fibrils, or are they going to dissociate back to monomers instead? And so for the amyloid beta 42 peptide that we looked here and under the conditions of the experiment, it turned out that the vast majority of oligomers actually do not form fibrils, but rather dissociate back to monomers. And so we were puzzled by this, and so we did some coarse-grained computer simulations together with Angela Saric to try to understand this mechanism at the molecular level. And these coarse-grained simulations, Angela will speak in a couple of weeks, so I'm sure she will give you some more insights into this type of simulations, but they're very useful because they allow you to consider experimentally measurable quantities while retaining molecular level resolution. And so very briefly, in these simulations, the protein is modeled as a single particle that can exist in various states, and then it can convert between these different states: a soluble state, an intermediate oligomer forming state, and a fibril forming state, which has an interaction laterally here, which allows to form fibrillar structures. And then what we see in the simulation, exactly like in the experiments, is that the vast majority of the oligomers that form on the fibrils are dissociate back to monomers, and that very occasionally they form new fibrils. And so we ask, why is this in the simulations? So this is because larger oligomers have a larger probability of converting. And so effectively what happens is you have a structure that keeps changing interactions. You have to nucleate a subcluster of a sufficient number of stabilizing beta sheet interactions in order to for this new fibril to grow. And so if this conversion doesn't happen in time, then these oligomers are going to dissociate back to monomers. And this type of mechanism predicts that the percentage of converting oligomers increases with concentration. And this is in fact what we see with the experiment. And this is because at higher concentration, not only we have more oligomers, but these oligomers are larger on average. And so they have a higher probability of converting, and so of undergoing self-replication. And so what we see here from this data and also theory and simulations is a two-step effectively nucleation mechanism for these oligomers, or for these fibrils, via formation of these intermediate oligomers, which are largely disordered structures that keep changing shape and interactions. So now that we've learned something about the mechanism, we can turn back to our question of controlling this process with inhibitors. And so what we've learned, for example, in the context of the amyloid beta peptide, is that there is this primary nucleation step which forms the initial fibrils, but then after a certain critical concentration of fibrils is formed, the main driver of amyloid formation is this autocatalytic loop mediated by secondary nucleation. And this gives rise to this exponential growth in the initial stages of aggregation. And this is the regime where we are going to work. So we are going to focus on the early stages of aggregation, where the aggregate concentration increases exponentially with time. And so we can capture this situation with a very simple linear differential equation, which has a multiplication rate kappa 0 and an injection rate alpha naught that comes from primary nucleation. What happens if we now add the drug, which we can view now as our control variable that we can tune from the outside to drive the aggregation reaction in different directions, and in particular reduce the concentration of toxic aggregates. So in the context of a drug, this drug could bind various species, either the monomers, the oligomers, the fibril ends, or the fibril surface, and in this manner affect various or all of the steps in the aggregation. And it turns out that if the binding of the drug to the target is fast compared to aggregation, then you can describe the kinetics of aggregation in the presence of the drug using the same equation as in the absence of the drug, but the rate parameters, the alpha and the kappa, are now functions of the drug concentration. And the specific form of these functions here depends on which species is targeted. Now, to close our control problem, we have now to specify a cost function, which in this context should penalize aggregate concentrations and high concentrations of the drug. And so in the simplest setting, we can set up a cost function, which is linear, both in the aggregate concentration and in the drug concentration. And this parameter zeta here is a relative toxicity that measures the relative toxic effects of the drug as opposed to the aggregates. And now we can use the tools from control theory to find the optimal protocol for giving this drug, depending on the mechanism of action. And so here is the solution to this control problem. So what you see here is that because of the linear nature of the cost function, the best protocol is actually a bang-bang, as we have seen for the saline problem. So it consists of constant level of the drug given for a certain amount of time. And what is interesting here is that the protocol is fundamentally different depending on whether the drug inhibits primary nucleation or it inhibits secondary nucleation or growth. In the first case, you have to give the drug right away for a certain period of time, and the duration of the treatment depends on various variables such as the drug concentration and also the strength of binding. In the context of an inhibitor of secondary nucleation or elongation, there is a waiting time before you give the drug. And so we were puzzled by this possibility to link, if you want, mesoscopic protocols for giving the drug with the underlying molecular mechanisms. And so I went back to my collaborators in Cambridge who work on a C. elegans model of Alzheimer's disease to test this prediction. And so in this model, they are considering C. elegans worms that can be made to express human amyloid beta in their muscle cells. And so as amyloid beta then aggregates in the muscle cells, the worms experience paralysis over time. And so we can look, for example, at the frequency of body bends or some other measure of mobility as a readout of the progression of the disease. And then you can give different drugs to see how this toxicity is rescued. And so here we are considering data for bexarotene, which at the molecular level has been shown to inhibit primary nucleation. And so from the theory, we would predict that an early administration would be more effective than a late administration. And in fact, in the data here, we see that an early administration is much more effective at rescuing the frequency of body bends than a late administration. And in this context here, we are considering another drug which inhibits secondary nucleation. And so in this case, we see that actually a late administration is much more effective than an early administration, in line with what is predicted by the theory. So this is good because it works, but of course it's just a proof of principle. And in the future, it would be very interesting to include other effects such as metabolism or stochastic effects coming, for example, from measurement noise. And incomplete knowledge, for example, of the link between aggregation, for example, and your readout of toxicity. Now, so this was a strategy to think about controlling aggregation in time using drug, but an orthogonal strategy could be to think about spatial control. So we would say, for example, can we use liquid condensates as a way to localize the formation of these aggregates in particular regions, for example, in a cell? And so here, of course, I'm thinking about liquid condensates or membrane-less organelles, which have emerged recently as a new organizing principle for biochemistry inside cells. So these are liquid droplets, or very similar to liquid droplets. They are ubiquitous inside cells, and they form as a result of stress. They concentrate genetic material, proteins, and in general, they provide different biochemical environments for performing and affecting chemical reactions. And so an interesting question could be to ask what happens if we have a liquid condensate that could localize the formation of these aggregates. So what, for example, what are the physical principles or the physical parameters that determine the partitioning of the aggregates inside these condensates? And so to answer this question, I have combined the kinetics of protein aggregation with liquid separation. And so to give you a little bit of an introduction, let me very briefly review the basics of phase separation. So let us consider the situation where we have a system of volume V, which contains a phase-separated condensate. This condensate forms as a result of phase separation between two components, A and B. A is the droplet material, could be some protein material, and B is the rest, so cytoplasm or nucleoplasm, and it could be water, lipids, proteins, or some mixture of those. The key parameter in this context is the volume fraction of A, which of course, because of incompressibility, is also related with the volume fraction of B. Now, the minimal or the simplest model for phase separation comes from Flory-Huggins, which effectively considers the phase separation to be the result of two opposing effects. On the one side, the fact that if you have these two components, A and B, they want to mix. And this is in order to maximize the mixing entropy, and so the number of configurations that these molecules can adopt. But of course, we have also the interaction between these molecules. And if these interactions, for example, are sufficiently disfavoring between A and B, then this interaction can drive phase separation and overcome the natural tendency to mix. And this interaction is described here by this Flory-Huggins interaction parameter, which is a mean field parameter that takes into account self-interactions and interactions between A and B. And so for a certain value of this interaction parameter, you can have in a phase diagram the situation where you can phase separate. And so in this context, you can then use this model to calculate the volume fraction of the droplet material inside in the dense phase and in the outside in the dilute phase. So now let us add the monomers into play, which are considered to be the clients in this reaction. So A and B, A is, if you want, the scaffold of the condensate, whereas the monomers are clients which are hosted by these condensates. And the idea is that these monomers are not going to affect the phase separation of A and B, but they are going to partition inside or outside depending on their relative interaction between the inside and the outside. And so this can be described by a ternary Flory-Huggins, which has terms for the monomer mixing on top of the mixing parts for the A and the B component. And also we have to add interaction terms for the monomers with A and for the monomers with B. And we are going to look at the limit where the monomers are dilute in the system. For example, for typical nanomolar concentrations and typical size of proteins, the volume fractions tend to be very small. And so this allows us to expand the free energy for a small volume fraction of the monomers. And in this context, you can calculate what is the partitioning of the monomers, which is effectively the ratio of the concentrations inside versus outside. And so as kind of we imagined, or our intuition told us, what results in this context is that the monomers are not going to affect the phase separation, but they are slave to the phase separation. So they are going to partition inside and outside, depending on the relative interaction with the inside and the outside. And this is what you see here from the partitioning, which turns out to be an exponential of the net monomer interaction, which is effectively the difference of interactions between the monomers and B and the monomers and the A material. And so depending on whether the monomers like the inside more than the outside, they are going to partition mostly inside or outside. And also the degree of phase separation plays a role in here. And so at the critical point, the curve is flat. And as we depart from the critical point, we get these exponentially growing enrichment functions, which, because of this exponential term here, would give rise to relatively large enrichment factors for relatively weak interactions of just a couple of kBt. So let us now add this equilibrium partitioning of the monomers with the aggregation reaction, which will drive effectively the system away from equilibrium. And so I'm going to consider a situation where we have a separation of timescales between the diffusion of monomers, which is supposed to be fast. For example, for a protein on the length scale of a cell of a couple of tens of micrometers, the diffusion time could be in the order of seconds, whereas typical timescales for aggregation in experiments are hours or even longer. So we have this timescale separation. And also I'm going to neglect the diffusion of the aggregates, which because of their size experience a much larger drag force and could also get stuck if they form some kind of mesh-like structure. So what is the consequence of this timescale separation between fast monomer diffusion and slow aggregation? Is that effectively the partitioning of the monomers is going to stay close to equilibrium at all times. But of course, as aggregation happens inside or outside of the condensate with different rates, this will create an unbalance. So some monomers are going to be consumed, for example, in the condensate, and this will create an unbalance in the partitioning, and the system will try to reestablish the partitioning in equilibrium. And so from the linear response theory, we know that this means that there will be a diffusive flux between the inside and the outside that will try to maintain the partitioning of the monomers close to the equilibrium value at all times. And so this intercompartmental flux is, as expected, dependent on the deviation of the monomer partitioning from the equilibrium value. So when the monomer partitioning equals the equilibrium value, then this flux is zero, as it should be. And so when setting up the kinetic equations of aggregation with a phase-separated compartment, we have to take two things into account. So on the one side, that aggregation will occur in both phases 1 and 2 as a result of growth, primary nucleation, and secondary nucleation. But also we have to take into account these diffusive fluxes that connect the inside and the outside, and that will try to reestablish partition equilibrium when aggregation will effectively take this away from equilibrium. And so let me show you the graphical solution of this set of kinetic equations. So how could a situation look like? So let us imagine we start with the situation where we have more monomers inside than outside because of the chemistry effectively of this. So we have a monomer partitioning larger than 1. Then because of the higher concentration inside, the aggregation is going to start inside of the condensate, and the first aggregates are going to form. Now, there is an imbalance of the monomer partitioning between the inside and outside, and so there will be a diffusive flux of monomers from the outside that will try to reestablish the equilibrium, and that will give more monomers inside, and so the aggregation inside is going to be accelerated, and that will draw more monomers into the inside, and so on and so forth. And so you can see that there will be a positive feedback mechanism, which is mediated by the phase separation and a diffusive flux, which could ensure that the vast majority of aggregates form inside of the condensate. And so we can quantify this by considering the aggregate partitioning, which in this context I defined as the ratio of the final concentration of aggregates that are formed inside of the condensate versus outside. And we looked at this as a function of the monomer partitioning. And it turns out that this aggregated partitioning is a power law of the monomer partitioning, where the exponent depends on either the reaction orders of secondary nucleation and growth for small condensates or primary nucleation for large condensates. But in either case, this means that this initial monomer partitioning is going to be amplified because of this positive feedback to a large aggregate partition. So for example, for the amyloid beta peptide from the in vitro experiments, the reaction order for secondary nucleation is close to 2. So that means an exponent of 3. So that means the partitioning, monomer partitioning of 10 is going to give rise to a 10 to the 3 partitioning of the aggregate. So with relatively weak interactions, this positive feedback mediated by the coupling between the inside and outside can give rise to strong aggregate partitioning. And so this is the end of the story. I hope that it was interesting and that I gave you a little bit of a flavor of how you can think about these processes from a control perspective. I would like to thank all my collaborators spread around the world, in particular Thomas Knowles, my PhD supervisor, with whom I did a lot of work on protein aggregation, and of course, all of you for your attention.
Thank you very much, Thomas, for the nice talk. Let's wait for a minute or so to see the questions. I have a question for you. It's a great talk. How do you account for the off-pathway oligomer formation in your theory?
Yes, so this is definitely something that can be done. So I haven't shown this in this context, but you can include effectively a species that is effectively an oligomer, but doesn't give rise to the fibrils. So as a little bit as a sink. And so in the context of the amyloid beta data that I showed before, it seems that the vast majority are not off pathway. But of course, the situation can be very different as you change concentrations and the conditions. And so I think it would be, yes. So I would say for this specific experiment, this is what we found out. But I would be very careful about making broad claims out of this, particularly because other people have also noted these off-pathway oligomers under different conditions. But in principle, this is a framework that can be applied to address this type of question. In particular, we have done some work with Alexander Deer, a postdoc in Thomas's lab, who has actually really gone very much in detail in this question of how to detect from a kinetic perspective these off-pathway oligomers. So I definitely recommend looking at these publications on this.
Participants, please post your questions in the Q&A folder. And the panel members, you can post questions in the chat folder or raise your hand. I have another question. So this theory works nicely when everything goes well in an ideal sample condition. What happens like the external forces like mechanical agitation or electric force? Can you account for that as well? in the theory?
It's a very good question. So I guess I've been very lucky to work with amazing experimentalists who can provide very nice data. So I guess, these are effects that can be taken into account. So for example, there has been some work also from Thomas's lab and Sarah Lindsay, who has looked, for example, at the role of agitation in the context of amyloid beta. And so how increasing, for example, the agitation rate gives rise to effectively fermentation in the system. And so you can see very nicely from those data how the dominant mechanism shifts from being secondary nucleation to being some more related with fragmentation. And it's very nice because this is something very technical, but actually it's quite interesting. And it's associated with this generalized logistic function here. Depending on the mechanism, whether it's fragmentation or secondary nucleation, the shape of the curve is very different. So when you have secondary nucleation, the takeoff is way more curved than the approach to the plateau, and the opposite situation happens with fragmentation, which has a slow takeoff and a very rapid curve here. And so you see very nicely in those data how the curve shape switches in terms of curvature and becomes more close to fragmentation. And so again, this is an example of an experiment with controlled, external, mechanical forces, which can be included in this. And so in principle, this could be studied.
Okay, Danilo, go ahead.
Okay, I think we are a bit late, so a very short question. And I wonder, is there great talk? I enjoyed very much. I wonder, is there any relationship between aggregation mechanisms and propensity of the aggregates to be degraded by proteases? I mean, the beta sheet content, in other words.
I guess, yeah. So I'm not familiar very much with these things. So I have to admit, I don't know very much. But there is some work that I'm going to start with some collaborators here at ETH who are looking at reversible amyloids, so which are effectively potentially degraded by some chaperones or things like that. And so this we are going to model with the similar techniques by including effectively inhibitors, which would would, for example, promote the dissociation rate, which I have included into here. So for most of the conditions that we looked at, the fibers are very irreversible. So effectively, there is almost no backward rate, except for the oligomers, which are very kind of closer to the monomers in some sense than to the fibrils. But then once you pass this kind of barrier, then everything becomes super irreversible. So the dissociation rate is very small. But of course, for other systems, so for example, actin and tubulin, the situation is different. So this process becomes much more important. And for example, in the presence of chaperones or so, they could, for example, enhance this by active mechanisms. But personally, I'm not very familiar whether there is a direct connection of this. I would say, the ratio, for example, between the forward rate and the backward rate, it would be a kind of readout of how irreversible effectively the system is, and so how difficult it would be to somehow revert this back. But this would be my guess.
Okay, Thomas, thank you very much. In the interest of time, let's take these questions in the Q&A folder at the end. So now let me hand over the podium to Dr. Abideni, who is going to introduce next speaker, Dr. Raleigh. Go ahead.
Thank you, Rams. So it is my great pleasure to introduce Professor Raleigh today. Dan received his undergraduate degrees in mathematics and in chemistry from the California State University system. He received his PhD with Dr. Robert Griffin at MIT, working on the theory of solid-state NMR. He then moved to Oxford as a postdoctoral fellow in the laboratory of the late Dr. Christopher Dobson. He returned to the US as a visiting scientist in Dr. Bill DeGrado's group, then at DuPont. Professor Raleigh began his independent career at the State University of New York, where he is a member of the Laufer Center for Quantitative Biology and professor of chemistry. He is also professor of biophysics in the Institute of Structural and Molecular Biology at University College London. In addition to his work on islet amyloid, his lab studies unfolded states of soluble proteins, develops methods for the design of hyperstable proteins, and studies protein membrane interactions. He has received a range of awards for his work, as well as multiple teaching and mentorship awards, and is a past president of the Protein Society. Now, without further ado, I will turn it over to Dan to present his talk today entitled Amylin-Induced Beta Cell Death, from Molecular Biophysics to Potential Therapeutic Applications.
Great. Thank you very much, Andy. And let me begin, first of all, by thanking Joan, Magda, Rams, and Sam for putting together this seminar series that we've all been enjoying over the last couple of years, and also to thank them, of course, for the opportunity to speak today. I'm going to give you an overview of some of the things that we're interested in and rather than go into a lot of detail, I'll try and just hit a few things that we're currently focused on. Just by way of formality, let me go through the NIH-mediated disclosures, which aren't particularly interesting, and let's move on to the more interesting stuff, which is islet amyloid formation. There's a long history of islet amyloid formation, actually predates Alzheimer's. Famous paper, ours, goes back more than 120 years to a paper by Opie, who reported on the relationship of diabetes to lesions, i.e. plaques, in the pancreas. This work lay fallow for almost 60 years until Irving Ratner at the New York Blood Institute positively identified them as amylin, and the natural assumption at the time was that these were deposits of insulin. That turned out not to be the case, but it wasn't until 1987 in a series of very important papers from the Westermark lab and from Ann Clark's lab that the deposits were shown to be composed of a new polypeptide hormone, which is now known as amylin, and also referred to as islet amyloid polypeptide, or IAPP. As I'm sure most of you know, it's a 37 residue polypeptide. It has two key post-translational modifications. There's a cysteine-cysteine disulfide bridge linking residues 2 and 7. There's an absolutely conserved amidated C-terminus. Physiologically, the peptide plays a role in glycemic control. It's synthesized in the pancreatic beta cells, stored in the same secretory granule with insulin, so by definition will be released in response to the same stimuli. Its primary functions are to slow gastric emptying, to promote satiety, and it's also believed to inhibit the secretion of glucagon by the alpha cells. However, in the pre-type 2 diabetic state, amylin starts to aggregate to form islet amyloid, and this process is deleterious to beta cell function, ultimately leads to beta cell death, also contributes to the failure of islet grafts, islet transplants. And you can see that on these images here of human non-diabetic tissue, pancreatic sections stained for insulin and for amylin, no detectable amyloid. In contrast, from pancreatic section from someone who had type 2 diabetes at the time of death, there's less insulin positive area and much more positive amylin area. Human amylin is one of the most amylogenic polypeptides known in vitro. It's aggressive, aggregation prone, more so even than the A-beta peptide. There's an interesting correlation between the ability of an organism to develop diabetes and the type of amylin that it makes. And we'll be coming back to this later on in the talk, so let me just mention it now. There's essentially a one-to-one correlation between organisms which develop diabetes and form islet amyloid, they secrete a form of the polypeptide which is very aggregation prone. Species which do not develop diabetes do not form islet amyloid, and they secrete a form of the peptide which is non-amylogenic, at least under normal conditions. It can be forced to be amylogenic under very high concentrations or under forcing conditions. Amylin, like many polypeptide hormones, is made as a pre-pro sequence. Signal sequence is cleaved off early, and then their processing signals at the N and the C terminus of the molecule, which are rigorously conserved. I'm not going to say much at all about the processing at the N terminus, simply a remark that it involves prohormone convertases, which cleave this dibasic signal, and point out that there is good evidence that a misprocessed form of the polypeptide is secreted in diabetes, which involves this N-terminal extension. The C-terminal processing seems to be okay. There are some issues with the N-terminal processing. This has been reported by a number of groups. We've recently shown that it activates the amylin receptor. It is toxic to beta cells, and it's known that it will aggressively interact with components of the basement membrane, and that can accelerate aggregation. The C-terminal processing is also strictly conserved and is quite interesting, a bit more complicated. Again, a dibasic sequence, which is the recognition motif for the prohormone convertases, that trims off most of the C-terminal extension. Those two dibasic residues get cleaved by a carboxypeptidase, leaving a glycine, which acts as the nitrogen donor for amidation. And the amidation signal is rigorously conserved among all known amylins. And in fact, the enzyme that does this, so-called PAM, is also found in all studied vertebrates. So why amidation? Well, it turns out it's important for function. Amylin is a member of the so-called calcitonin peptide superfamily. It does bind to the calcitonin receptor, albeit with less affinity than calcitonin. But the main receptors are these three, which are more specific for amylin. They involve the core of the calcitonin receptor, are complexed with these interesting so-called RAMP proteins, receptor activity modulating proteins. And in the binding pocket, there are a number of acidic residues. When the amylin docks, that N-terminal amide group forms hydrogen bonds. So simple electrostatic repulsion would lead to the prediction that a molecule with a free C-terminus would interact much more weakly with the receptor. And in fact, the very simple change of replacing the amidated C-terminus with the more common carboxylate leads to a 60-fold reduction in receptor activation. And to put that into perspective, that is significantly larger than has been observed for any point mutant of amylin that has been characterized. There are a number of oddities about amyloid formation by amylin, which are kind of fun to think about and worth mentioning because they may come to play a role in other systems as well. And the first that I want to mention is that amyloid formation is strongly dependent on the choice of buffer, actually on the choice of anion. One of many examples is shown here. A number of years ago, we were interested in a variant des-lys amylin, which lacks the N-terminal lysine, and there's some thought that this species could be circulating. We've shown that it's perfectly good at activating the amylin receptor. In tris, this deletion of the lysine leads to more rapid aggregation, quite easy to rationalize. As amylin, if you look at the sequence, has no acidic residues, and remember that C-terminus is amidated, so at any physiologically relevant pH, it's going to be cationic. In fact, the highest density of charge is lysine 1. So removing lysine 1 should reduce charge repulsion and lead to faster aggregation. what we observe. In contrast, there's no observable effect in phosphate buffered saline, and it's due to the phosphate, not to the saline. And what's going on here is we're still diminishing the charge repulsion, but we're also weakening the interactions with phosphate. Phosphate is known to interact reasonably well with amylins, so we have two counteracting effects. Reducing the charge accelerates aggregation, reducing the interactions with phosphate slows it, and they more or less cancel each other out. We've observed similar effects for multiple mutations and variants that involve the addition or subtraction of charges. In fact, there's quite a rich spectrum of anion interactions with human amylin. That's shown here at low anion concentrations. These scale as the classic anion selectivity series, at higher concentrations as the Hofmeister series. As thiocyanate provides a particularly interesting example. That's commonly viewed as a very strong protein denaturant, yet at low concentrations, it significantly accelerates amyloid formation. At high concentrations, it significantly retards it. Another amusing feature of amyloid formation by amylin is there are examples of thioflavin T negative amyloids. This is one of them, taken from some work we did with Joan Emichet's group a number of years ago. We were interested for a variety of reasons in fish amylins. And this is the thioflavin curve for the human peptide, what we're all used to looking at. The thioflavin curve for the fish peptide is flat, but if we take aliquots and record TEM images, we see lots of fibers, and these are rich in beta sheet as judged by CD and IR. So I mentioned amylin is found in all known vertebrates. There appears, however, to be absolutely no selection against amyloid formation. In fact, of all of the possible amylins, our lot seems to be to have gotten just about the worst possible sequence. If you look at predicted ancestral sequences generated from ancestral sequence reconstruction, they're all less amylogenic than the human peptide and less toxic to pancreatic beta cells. If you look at amylins from species which are currently alive, but evolutionarily very distant, the same is true. These two bizarre looking things are the latimeria, the vertebrate which is believed to have the genome which changes the most slowly, and this even more strange looking thing, the so-called ghost shark, is essentially among the vertebrates the farthest removed from us evolutionarily. Hey, they make amylins, and their amylins are much less aggregation prone than ours. It's natural to ask, why do we have such a bad amylin? What are the toxic species that are produced, and what are the mechanisms of toxicity? And I stress mechanisms because there are certainly more than one. This experiment illustrates that toxicity resides in the lag phase for amyloid formation. In these experiments, the human polypeptide is incubated for different amounts of time and then added to cultured INS1 beta cells and cell viability read out. You'll notice that at species that are formed very early are not toxic. Those in the midst of the lag phase are, and the fibers are not. And again, I stress these experiments are done where incubation is done separately on the bench. The species are characterized and added to cells. It's not that we're adding peptide to cells and looking at what goes on continuously. We've also found with other amylins from higher primates, baboons in particular, our fibers are not toxic. The only known mutations in human amylin that are disease-linked, also the fibers are not toxic. The A's prefibrillar oligomers do a number of nasty things. They upregulate pro-inflammatory cytokines. They enhance ROS production. They lead to elevated caspase-3 cleavage. And recently, we've shown that they lead to breakdown in membrane asymmetry in the beta cells. So if the fibers are really not toxic, and these pre-fibrillar species are, we should expect to observe a correlation between the length of the lag phase and the time over which a particular polypeptide remains toxic. We can test that conjecture because we have a large number of mutants in the freezer. Some examples are shown here. The S20G mutant is particularly interesting. It's found in nature. It's linked to a slightly higher risk of type 2 diabetes in a small subpopulation of individuals of Japanese and Chinese origin, forms amyloid faster than wild type. On the other hand, the S20K mutant significantly more slowly. So we have collection of mutants and studied the human protein under a range of conditions where we can vary the lag time by almost a factor of 500. And there's really this extraordinarily strong correlation, remarkable, between the length of the lag time and the time over which the species remain toxic. You may wonder about this S20GA mutant. As I said, it's linked to an increased risk of type 2 diabetes, but it aggregates faster, and we're telling you that the amyloid fibers are benign. Well, it turns out that the oligomers that are formed by the S20G mutant are more toxic at their time point of maximum toxicity. We also believe there's a role for loss of function. We've shown that it leads to decreased activation of amylin receptors, and in preliminary data collected in collaboration with Thomas Lutz at the University of Zurich, this S20G mutant is less effective in controlling appetite in rodent models. There are a tremendous number of studies of molecules which modulate amyloid formation by A beta and amylin. I'll just mention one, the non-steroid anti-inflammatory flumiprophan. And we discovered fortuitously that it accelerates amyloid formation by amylin, but it also reduces significantly toxicity towards cultured beta cells, and it also protects transgenic islets, which produce human amylin. And the idea here is we're simply reducing the population of these toxic oligomers. What can we say about the toxic oligomers? Well, they're clearly heterogeneous. They contain partial helical structure as judged by CDA, infrared spectroscopy. These are two-dimensional infrared experiments collected in real time with our collaborator, Martin Zanni. They reveal partial beta sheet structure. This in situ cross-linking reveals a family of oligomeric species. And I just want to comment that this is completely consistent with ion mobility studies that were pioneered by Sheena Radford and her colleague, Alison Ashcroft. The aromatic residues in amylin are still largely exposed. We know that from work with cyano-Phe analogs. These prefibrillar species don't bind any of these polarity-sensitive dyes, unlike some other toxic oligomers. Let me just mention this one, diphenylhexatriene. Those of you who work on membranes may recognize it as a probe of membrane fluidity. It turns out it's also an extremely sensitive dye for amyloid formation, at least for amylin, and reports faithfully on the kinetics. So we've started to make a lot of use of this. One does need to be a little careful because it tends to bind to plastic surfaces, but it is extraordinarily sensitive. What about mechanisms of toxicity? What is the biology behind these deleterious effects? One important mechanism involves interactions between amylin and a pattern recognition receptor that goes by the name of RAGE. Andy Abedini's laboratory showed that RAGE is upregulated in transgenic mouse models of islet amyloidosis, those that express human amylin. You can see that here in the immunohistochemistry. Blocking the interactions between RAGE and amylin diminish toxicity. That can be done either by an anti-RAGE antibody or using transgenic mice that have RAGE knockouts. In both cases, there is significant, albeit not complete, protection against toxicity. Right. In the remaining few minutes, let me turn to a practical problem. The question that we're asking is, can we build a better amylin? Soluble analogs of amylin are of interest for the treatment of diabetes, but particularly for obesity. There is one molecule that's been approved by the FDA, known as peramylintide, marketed under the name of Amylin, and it's based on a simple sequence comparison of human amylin and rat amylin. And what was done was the 3 proline substitutions in the rat polypeptide are substituted into the human polypeptide, and this was approved back in 2005 for the treatment of diabetes. It's used, but it's not used extensively. More recently, there's been really impressive progress, particularly by Novo Nordisk, on analogs of amylin that can act as anti-obesity agents. Amylin and the current molecules still suffer from poor solubility at pH 7.4. Peramylintide needs to be formulated at acidic pH. That's an issue if you wish to co-formulate with insulin, because insulins are formulated at pH 7.4 or slightly higher. So our goal is, can we develop bioactive, non-toxic analogs that are more soluble than peramylintide at pH 7.4 and could be co-formulated with insulin? The ultimate goal of this would be to design an artificial pancreas that could secrete insulin on an amylin analog and glucagon. So our strategy has been to use the minimum number of prolines and the minimum number of charge substitutions, and we want to put them as close as we can to critical region for amyloid formation. But what is this critical region? How do we proceed to aid? This slide summarizes a very long collaboration with Martin Zanni's lab, and I won't go into any detail except to say that we use 2-dimensional infrared spectroscopy, with specific isotope labeling, which has the advantage that it's an in situ, non-perturbing technique, significantly more sensitive than linear IR, and one can collect spectra in real time. The key point for us today is it highlights the importance of residues 22 through 27 on this region of the molecule forms structure in these prefibril oligomers. This structure rearranges to go into a fibril competent state, and we believe that involves a significant free energy barrier and in part sets the lag time. From a practical perspective, it tells us what region to focus on. So the first question we will ask is, what about these rat prolines? Do we need all of them, and are they the best choice? Well, the answer to the first question is no, and the answer to the second question is also no. Some of the data in support of that is shown here. This is just an examination of the effect of these point mutants on amyloid formation, and you can see the S29P mutation really doesn't do much at all. No difference in the kinetics. Gobs of fibers are present. The A25 mutant slows amyloid formation but we still see extensive mats of fibers. Notice it has quite a weak thioflavin response, just reiterating the dangers of equating final thioflavin intensity with the amount of fibers that are formed. In contrast, serine 28 to proline does have a significant effect. Same thing is true if we look at the double mutants. Only those which contain a proline substitution at position 28 have a significant effect. And the same story holds through when we look at toxicity. Here we're looking at a cell viability assay, based on ATP production, and also another assay based on cell permeability. They tell the same story. A25 and S29P, they don't really do much at all. In contrast, our S28PA increases the amount of peptide one needs to add to reduce cell viability. The double mutants, same story, but actually these are a bit more interesting. And again, as long as we have an S28P substitution, magenta or orange, we see significant reduction in viability. Notice the 28-29 seems to have some synergistic effect, even though the 29 didn't have much effect on amyloid formation. So it is doing something to the toxicity. Now, sadly, the single and the double mutants, and in fact, the triple mutant, the Amylin, peramylintide, they're not very soluble. So what to do next? One of the really exciting things in the amylin field has been the publication of some cryo-EM structures recently from a variety of laboratories. The Schroeder Laboratory in Germany, Sheena Radford's lab in Leeds, and David Eisenberg's lab at UCLA. And David has also published structures of fibrils derived from human tissue. We've simply plotted ribbon diagrams of these with cross sections together with two earlier models based on steric zipper peptides, also from David's lab, and Rob Tico's model based on solid state NMR constraints. Well, they all look different. That's pretty obvious. That highlights the polymorphic nature of amyloid formation, particularly by amylin, but they do have some things in common. And one thing they have is glycine 24 forms tight contacts in all of these structures. So we were interested in what would happen if we replaced this glycine with larger residues. So Lakshan and Rahana in the lab used natural amino acid, 2-aminobutyric acid, and leucine. And in both cases, we predict based on energy decomposition within the context of the amber force field that these will be significantly destabilizing, particularly for this leucine substitution, yet both of these substitutions significantly enhance the rate of amyloid formation. So it's very difficult, at least for us, to predict what mutations will do based on existing structures. It's also difficult to predict based on algorithms. There are a number of the A's that have been developed over the years, some quite successful. They work for some substitutions in amylin, but not for all. So the in silico methods don't work particularly well either. The alternative strategy that we have adopted is to examine amylins from organisms that we know do not form islet amyloid, or from organisms that face metabolically challenging environments. Domesticated pigs, domesticated cattle, and domesticated sheep do not form islet amyloid. Cattle do form a type of diabetes, but it's believed to be viral in origin. This is a North American brown bear. It's the largest apex predator in North America. These things are very good at gaining weight. They essentially triple their weight before hibernation. They do not form islet amyloid. They do not develop diabetes. So we examine on the amylins from these species and observe some rather interesting effects. We'll consider just the bovine variant in the interest of time. There are a number of interesting substitutions: histidine 18 to proline, a couple of asparagine to lysines. This molecule is non-amylogenic. It's extremely soluble. It's not toxic. Unfortunately, it's worthless for any therapeutic applications because it doesn't engage the human amylin receptors. That's largely due to the histidine to proline substitution plus a few others. Strikingly, these asparagine to lysine substitutions have no statistical effect. And notice N22 and N31 are very close to this core region. So our strategy was to take advantage of lysine and substitutions for asparagines. I should have mentioned this is a common theme also found in the sheep and in the bear. and in the pig peptides. We've made a number of variants, 11 in total. I just show you 8 here. The important result is they don't form amyloid. These are thioflavin experiments, black for the wild type human, color-coded for various analogs. They're also not seeded by the human fibrils. How many prolines do we need? Well, we need at least 1 with the designs that we've worked on. We need at least 2 charged residues, additional charged residues. We focused on asparagines for the reasons I had mentioned. Doesn't matter if it's lyse or arg. And interestingly, and to be honest, quite surprisingly to me, in some cases, glutamic acid works just fine, even though that reduces the net charge on the molecule. So there's no one-to-one correspondence, no monotonic relationship between net charge and solubility or amylogenicity in these molecules. What's particularly encouraging is these analogs are not toxic, and one of them, the one highlighted here in magenta, is as active as the wild type human peptide. This molecule includes the 2 asparagine to lysine substitutions that we stole from the cow, plus a proline at position 28. These molecules are also considerably more soluble than human amylin or Similin in the context of formulation with insulin. We made no attempt whatsoever to optimize the formulation. We simply used a commercial insulin under artificial challenging conditions, namely 1-to-1 with amylin. Therapeutically, you would give amylin at a much lower dosage. But the key result is within less than 24 hours, you can detect no soluble human amylin. Within a week, the Similin is gone. Up to at least 30 days, we see a significant amount of our designed peptides hanging around. And I'll just end by pointing out that we think we can do better. This is the result, the partial results from a proline scan of amylin on a plot of the T50 for amyloid formation, and you can see that there are certainly substitutions which have more drastic effects than S28. Some of the AAs are not useful for us because they significantly reduce activity but there's clearly a wide spectrum of peptide space to play with. Now, I think I've probably run over by a minute or two, so let me switch to the most important slide of all and thank the people who actually did the work. In London, Alex has been key with absolutely everything, and he guided our work on amylin evolution. On experiments were carried out by 2 students, Aria and Yvonne. Alex also contributed to our work on the proline scanning experiments and the design of the soluble analogs. Rahana and Matt in Stony Brook were the main players in the soluble analogs. Zach, Matt, and Dawn worked on the proline scanning. Minghao primarily works on the interaction of amylin with asymmetric membranes, but he did the work on the dye binding, particularly the DPH. Lach Ann's project is focused on another area, but he did the energetic analysis and examine the effect of the small to large substitutions. I've tried to mention our key collaborators as I've gone through. Let me just highlight them here. And let me finally finish by, again, special thanks to Rams, Magda, Sam, and Joan for organizing these seminars. And thanks, of course, to all of you for your attention and your patience, and be more than happy to answer any questions.
Great talk, Dan. I liked it very much. Could you comment on the observation that the very early oligomers, you mentioned that they are not toxic, but the oligomers formed later were toxic. Could you comment on that? Did you say that? Am I correct?
Beyond the phenomenological observation on and it's difficult for us to say anything. And with the human peptide, we see that it does take some time to develop toxicity. There's some mutants, particularly some of the aromatic mutants, which take a very long time to form amyloid, and they take quite a while to develop toxicity. But at this point, we can't associate that with any distinctive structural features. Would like to very much, but as of yet, we can't tell you any molecular reason for that.
And you mentioned about the bovine amylin to be pretty much useless. What is there any, why is it being produced by bovine then? There must be some reason, right?
Oh, I'm sorry. So it is useless to us, but presumably it's very useful to cows. The amylin receptors for cattle don't have a really strong sequence identity with the human receptors. So it was a bit of an anthropomorphic statement.
Okay. Binzu, you have a question? Go ahead.
Yes, hi, great talk, Dr. Raleigh. I have a question related to the T-half. So you have a collection of a mutant amylin. A number of them have a much prolonged T-half. Have you tested them whether they are less toxic or most of them are less toxic? And related to that question, I think you have done lots of inhibitors, small molecule inhibitors. Whether do you find also any like inhibitor like have a prolonged T-half leads to the aggregation significantly longer T-half to reaching aggregation?
Yes. So in answer to your first question, some of the mutants that we've characterized that take a very long time to form amyloid, they do take longer to develop measurable toxicity, but the peak in toxicity, as it were, the time point where they're maximally toxic is noticeably reduced compared to the human peptide. Probably the most striking example of that is a triple aromatic to leucine mutant, which forms amyloid very slowly, and it is toxic, but at any given time point, it's less toxic than the human's peptide. On the other hand, if you added it to cells and came back 3 days later, where you're sort of looking at integrated toxicity, you would see significant toxicity. And then your second question about the inhibitor, we have over the years, like many laboratories, found a number of small molecules that inhibit amyloid formation in the sense they either lengthen the lag phase considerably or they abolish the ability to form amyloid. EGCG, which your lab has worked on too, seems to be kind of a generic inhibitor of amyloids. That is pretty effective at protecting cultured cells. Some sulfonated aromatic compounds are in our hands. I mean, they'd be terrible drugs, but they work in cell culture. We have interestingly found some peptides, and this was work that was done on primarily by Andy Abedini. She found some mutants of IAPP which act as inhibitors of amyloid formation by the wild type peptide, but they actually prolong the time over which IAPP is toxic. We assume by binding to these toxic NATAs and slowing the conversion into the more benign fibril state.
Thank you.
There are questions in the Q&A folder. You can also take a look at them, so I'll read it for you. There's one from David Eisenberg. Dan, you mentioned the correlation of IAPP fibril formation in various species and the capacity to develop diabetes. Would you say that fibril formation of IAPP is a cause or the cause of diabetes type 2 in humans?
Thanks, David. That's a very good question. And we think, and I believe it's the general view in the beta cell biology field, that the process of islet amyloidosis is certainly a contributing factor, but it's a multifactorial disease, and there are other steps that are clearly involved. Peripheral insulin resistance is obviously critical, and to the best of my knowledge, islet amyloidosis is not linked to that. We focus mainly on type 2 diabetes because that's where the islet amyloid is, but there is now some evidence that in type 1 diabetes, there's some residual beta cell function, and islet amyloid could also play a deleterious role there.
Thank you. There's a question from Ada Capernuto. IAPP is known to bind insulin with high affinity. Did you study whether co-formulation of your IAPP analogs with insulin might affect insulin bioactivity or result in toxic co-aggregates?
That's a great question. And insulin is a classic inhibitor of IAPP aggregation on interactions of insulin and IAPP have been mapped in great detail, particularly by your lab, of course. We do not see the formation of any toxic co-aggregates. We have not tested whether that impacts the biological activity of insulin.
Okay, and related to that, you mentioned about the histidine 15, right? 18, histidine 18 to proline is a very unique mutant. Do you think its inability to bind zinc is playing any role on its function or toxicity or anything, any of those?
Well, it would certainly modulate zinc binding in vitro. I don't think that is critical, certainly not in the secretory granule, which is a pH 5.4 environment, so that histidine will be protonated, isn't going to be a ligand for zinc, and at least in the granule, most of the zinc is in the dense core in this semi-crystalline state with insulin, and when insulin is released, it's often released in a hexameric state. So I suspect not, but it is an interesting question.
Thank you. There are 3 questions from an anonymous attendee. For the toxicity of IAPP oligomers, does pore formation in the membrane play any role?
With cultured cells, yes, it does. What is the relative importance in vivo is very difficult to say. I think some of the experimental data that I showed you today illustrates that they're clearly non-membrane disruption-based mechanisms. Other mechanisms for which there are very strong evidence is activation of the inflammasome, work particularly from Steve Kahn and Bruce Fischer's laboratory. Lucy Marzaban, who used to be at UBC and has moved to Manitoba, has very strong evidence that IAPP oligomers bind to so-called death domain receptors. So there seem to be multiple receptor-mediated mechanisms, priming of the inflammasome, and probably also membrane damage.
The second question is, do you think we can learn something about the inhibition of IAPP aggregation by insulin?
Sorry, Rams, I couldn't quite catch that. Could you repeat that?
Do you think we can learn something about the inhibition of IAPP aggregation by insulin?
Yes, and I think that's not an area that we actively work on now. We used to. We did some work on the ability of insulin to inhibit these processing intermediates, showed it was considerably reduced in the presence of glycosaminoglycans, but it's not an area of our focus, but certainly other laboratories have done that. Insulin IAPP interaction sites have been mapped out in work from the Capernato laboratory, and there are a number of other studies. So the short answer is yes, but we're not engaged in that.
The third question is, what do you think about the LLPS in IAPP aggregation and toxicity?
So liquid-liquid phase separation, which connects nicely to the lovely talk given by Thomas. I'm agnostic. I don't think that there is any good biological evidence that it plays a role. There is still a debate in the literature whether IAPP oligomer formation and toxicity AA is dominated by intracellular or extracellular events. We fall in the camp that extracellular is important. I think Steve Kahn would agree based on his experiments, which link secretion on to toxicity. Certainly, there are rodent models where there's excellent evidence for our intracellular or basis of toxicity, but one needs to look closely. And in many of these, I think in almost all that I'm aware of, there are just tremendous number of copies of the human transgene, sometimes 30 to 50, and in many cases, it's under control of the human insulin promoter, which is a strong promoter. So they're producing, and their factories producing massive amounts of IAPP, and certainly in those cases, ER stress and other things clearly play a role. Whether that plays a role in a more physiological environment is an open question.
The last question for you is from Burgess and Velayudham. Is there any role for viscosity on amylin amyloid formation?
In vitro, yes. In vivo, the short answer, I don't know. And this came up in part, and I'll just mention this because I think it may be of some interest to Rams. We've been very interested in looking at asymmetric membranes, and the traditional method for making those involves encapsulation of vesicles in sucrose and separating by density gradient centrifugation. That leads to massive osmotic imbalance, which of course leads to leakage. So we did a bunch of experiments where we thought, well, we'll just add sucrose outside of the vesicles, and then went and did control experiments. And under those, so likely viscosity. The person who's done the most work on that, largely in the context of crowding, is Roland Winter's lab.
Thank you very much, Dan. Thomas, are you there? There are two questions for you in the Q&A folder.
Yeah.
One is from
Sure.
Yuari. Very interesting talk. Have you tried to predict computationally a condition for aggregation in terms of concentration of a protein and its kinetics?
Yeah, so I guess this is a very good question. We have done some work with, for example, with Angela Saric, and I think she will probably talk about that in a couple of weeks in her talk, precisely about the question, for example, of fibrin self-replication, under which conditions it happens. And so what we found with the coarse grain simulations was that there was a narrow regime for self-replication or secondary nucleation to happen in terms of the protein concentration and the strength of binding of effectively the protein to the fibril interaction. And so you need the protein fibril interaction to benefit from the surface effectively to nucleate, but at the same time, if that interaction is too strong, you inhibit secondary nucleation. So that gives the kind of optimal conditions in terms of interaction and concentration for effectively this secondary nucleation, which is the main driver, for example, in this amyloid beta peptide to occur. So that's a good example for that.
The next question is from Prabhakaran Ram. A great talk for mechanistic perspective. What would be the impact of molecular crowding within the cell?
Well, I guess it's It's a very interesting question, very important. I think it's one of the key questions, I guess, in the future. For in vivo, I have to say it's very hard for me to say what could happen because there are so many things that are. But mechanistically, there can be various levels to this question. So on the one side, we know that some of these processes, like just elongation or secondary nucleation, can change the way they behave as we change the concentration. For example, there are saturation effects. So for example, elongation can saturate at higher concentration and become, say, concentration independent. And this is because they are intrinsically multi-step processes. And so whatever is the rate limiting step can change. And also there have been some more in vitro studies, for example, by Frank Ferrone and people like that, on the role of crowding, for example, on nucleation, for example, with non-polymerizing agents. And so in that context, it was shown that the role of this crowding is to effectively reduce these rates by several orders of magnitude. But then to translate this into in vivo, I guess it becomes very complicated also because there can be co-aggregation processes as well. And I guess it's a challenging question.
Thank you, Thomas. Dan, you raised your hand.
Yes, Thomas, I enjoyed your talk very much and was particularly interested in your treatment of liquid-liquid phase separation. I thought the Flory Huggins work was very nice and seems almost surprising that something that's been around for so long works so well. But that was meant as a compliment. I'm afraid it maybe didn't come out off that way, but I thought that was very clever. What and perhaps I missed this, but does the model also include mechanisms of resolubilization of the phase separated entities, so the dissolving of the droplets?
Yes, I think this is a good question. So I think, so in this model specifically, effectively the aggregating protein was a client, so it was not really having a feedback on the phase separation. So effectively the phase separation was more of a way to having effectively two compartments. But the situation becomes very interesting now if you couple the phase separation and the aggregation a little bit, maybe along the lines of what you were saying before with the previous question about the role of phase separation as a kind of intermediate. I think that is a very interesting question, because in that context, if the phase separating protein is the same that then aggregates, then there can be effects on the phase separation, and then, for example, it could be that you end up dissolving this condensate as you form these fibrils, and you are effectively consuming the phase separating material. I mean, from the theory perspective, it's an open area. There have been some very recent work by, for example, Christoph Weber in Germany, who has been trying to develop theories for chemical kinetics in the presence of condensates also. And I think, yeah, I'm very excited to apply these tools to such a situation, which might be actually relevant. And you were also saying about these oligomers as, what is the difference with the liquid? And I think it's, you had this, you showed very nicely how these oligomers are very diverse. They have very different interactions, and they try to kind of form this kind of cross-beta sheet architecture also. But, they have a liquid-like structure in some sense, but on a much smaller scale. And it turns out that potentially these liquid condensates in vivo might be also much smaller than they are in vitro. And so it's a very gray area, and I think it will be very interesting to see what these intermediates are, and whether some of the liquid theory can actually capture some of their features or not.
Great, thank you very much.
Well, and thank you for your talk. It was also very nice and interesting. And in fact, I wanted to ask, maybe if I can, whether you know something about the mechanism of these various drugs that you showed about, because there are different effects on the production of the oligomers and so on. So do you have an idea of whether they target different steps also in
We
the
have not
aggregation?
looked at that with the flubiprofen. In part, I mean, it's interesting. There are a couple other compounds which accelerate. The flubiprofen therapeutically wouldn't be very useful because taking large prophylactic doses of a non-steroid anti-inflammatory will have some obvious side effects. We are doing exactly what you suggest in collaboration with Tom Recker's group with the various anions and a number of other small molecules. And Tom has some very nice data he's generated over the years where he has effectively parameterized small molecule interactions with protein surfaces. So at least when you're in the Hofmeister style regime, and instead of using just the old-fashioned M value from protein folding, you can get a bit more atomistic detail, at least on the types of surface area. So that's where we're going in that direction.
Very nice, very nice.
Okay, thank you very much for the great talks, Dan and Thomas. And thanks, Andy, for joining us. And thanks to everybody for participating. Thank you all very much. Bye-bye.